Hello friends, Ace Botain. Next episode. In this episode, we'll be discussing cell, cycle and cell division, third part. So we can proceed. So I will share the screen so it will be convenient for you. I think screen came. So we can proceed. Cell cycle and cell division, third one. So what all topics we'll be discussing in this episode? Basically, 3 items, an introduction, then meiosis, significance, and as usual, question 5 theory and 5 NEET model question. So we start with the meiosis. The term came from Greek. So root word is meio, M-E-I-O. It means lessen, reduction, reduce. And that is how the name reduction division came. And 2 parts will be there. That is why double division came. Scientists concerned with the meiosis, initially studied by Van Beneden, Strasburger, Winnie Wharton. And this meiosis takes place in diploid cells or mother cells, that is 2N. And usually after meiosis, 4 haploid cells will be formed. And when we continue with meiosis, meiotic interphase is almost similar to mitosis. But here S phase will be a little bit prolonged due to phase is short or even absent in few cases. Meiosis has got 2 stages, meiosis 1, meiosis 2. First meiosis is called reduction division or heterotypic, not uniform. That is how the term came, heterotypic division. And second meiotic division is called equational, parallel to mitosis, or homotypic division. So reduction and equational, heterotypic and homotypic. I think that words are clear. Now, why meiosis need meiosis 1 and 2? Some reasons are suggested, which include 4 haploid cells has to be produced after meiosis. So normally after meiosis, 1 only 2. To get 4, 1 more. That is ordinary mitosis. That is why meiosis 1 and 2. Second reason, first meiosis can cause reduction in chromosome number, no doubt. But second meiosis will only cause DNA content reduced in each chromosome. Third reason, after crossing over 2 chromatids, have to separate. They're not identical now. They have to separate. Some suggestions why there is a need of second part in meiosis, that is meiosis 1 and 2. I think idea clear. Now, meiocyte cell which undergo meiosis, it is diploid. It's a regular question, 1 mark theory NEET question. Now, meiosis, 2 parts, reduction division and equational division, meiosis 1 and 2. Both of them have prophase, metaphase, anaphase, telophase, and cytokinesis. I think this page is clear. Now we can proceed to next one. Now, as usual, prophase 1. Prophase 1 is the more complicated event. In comparing with the mitotic prophase, it is very lengthy also. All comparing with the mitotic prophase, so more complicated event. And lengthy also, comparing with the mitotic prophase. And in meiotic prophase 1, there are 5 substages. That is only for convenience, 5 substages. They include leptotin, zygotin, pachytin, diplotin, diakinesis. So leptotin we can proceed. So when we get the meaning of the word, it will be clear. Leptotin is otherwise called leptonema. And what is the meaning of the term, the prefix part lepto? Lepto means thin. Tainia, all these stages we can apply tainia, T-A-I, not tapeworm, T-A-I-N-I-A means band-like. And leptotin can be otherwise called leptonema. Nema means thread. So that is why leptotin is otherwise called thin threaded stage, a common question. Chromosomal nature become more clear than the chromatin. Leptotic chromosome will be arranged in a special manner called bouquet stage. Here you can find just like a bouquet chromosome will be arranged. A feeling we can get. So it is called bouquet stage, that is meiotic prophase 1, leptotin substage, bouquet stage. That's the following type question. Now comes zygotin. Zygotin came from the term zygon, Z-Y-G-O-N, means yolk. Y-O-K-E, not the other yolk. Yolk or together. That is why zygo, together. second stage of phase 1, homologous chromosome, that is maternal-paternal, that is diploid. So homologous chromosome almost similar in basic aspects. But remember, one from father, one from mother. That's all. And this chromosome together, one from father, one from mother, they are called bivalents, two homologous chromosomes. Total 4 chromatids. So bivalence means 2 chromosomes, they are homologous. Total 4 chromatids, 2 centromere in a bivalent. So bivalent is same as homologous chromosome, one from father, one from mother. So 2 homologous chromosome. That we only take 2 homologous chromosome, bivalence. And in a bivalent, there will be 2 chromosomes, 4 chromatids, 2 centromere. This is a bivalent or tetrad that will be proceeding. Next, in chromosome A, we can find here chromatids 1 and 2. These 2 are chromatids of the chromosome A. And they are called sister chromatids. Same with the chromosome B, 3 and 4 are called sister chromatids. I think that 2 words clear, sister chromatids. And in zygote, that main events include pairing of homologous chromosome. That is why zygon together, pairing of homologous chromosome. And this pairing is called synapsis, famous question. And paired chromosomes are called bivalents. And in this stage, we can observe a structure called synaptonemal complex connected with the synapsis. Synapsis also called synthesis, SYN. Synaptonemal complex was present in old NCERT with the diagram, scientist, which organism discovered, etc. Presently in NCERT, the term only is synaptonemal complex. So total I given here in bivalence, 2 homologous chromosomes. In univalent or monovalent, single chromosome without pair. In monad condition, a chromosome with a single chromatid. In dyad chromosome with 2 identical chromatids. Here you can find. And that is after S phase. That is how 2 identical chromatids will be formed in dyad. I think the term's clear. That is bivalent, univalent. Univalent is otherwise called monovalent. Monad condition, dyad condition. And how many bivalents will be there in a cell? Diploid one in a cell. So number of bivalents in a cell is half the number of total chromosomes. I think that idea is clear. Now, next stage we can proceed is called patching. Patchy means thick. That is how the term, teen means thread. So this is a third stage of meiosis, 1 prophase. Synapsid chromosome, paired chromosome will be more thick and short. So we can easily identify the tetrad condition. That will be very clear. This is the famous stage in which one of the important events, that is exchange of genetic material, takes place between paired chromosomes, causing variation. Many chemicals, enzymes, all involved in this procedure. That is technically called crossing over. I think that page is clear. Now we can continue with the crossing over. So again, paired chromosome, bivalence, synapsis or synthesis, A, B, A, 1 and 2 sister chromatid, B, 3 and 4 sister chromatids. So it is a bivalent, tetrad, 2 chromosome, 2 centromere, 4 chromatids. Crossing over takes place between exchange of genetic material between 2 homologous chromosomes between non-sister chromatids. So 1 and 2 are sister, 3 and 4 are sister. 1, 2 in comparison with the 3 and 4 are non-sister. So crossing over takes place between 2 homologous chromosomes, again between non-sister chromatids. A and B are homologous chromosomes. In A, 1 and 2, they are sisters, already I told you. In B, 3 and 4, they are sister chromatids. Non-sister chromatid can be compared with A and B. At the end of patching, the combination between A and B will be complete. And the paired chromosome will be linked at specific point where crossing over took place or taken place. Now comes next stage. It is called diplotin. Diplos means 2 or double. Diplo, we know, di. Here homologous chromosome separate except at certain points where crossing over took place. And that point is called chiasmata. it will be clear during this stage diplotin. And that area will be, our chromosome will be arranged like an X shape. Here the letter you can find X shape. So that itself was a question. And in the developing egg or oocytes of some vertebrates, diplotin lasts for even months. Thus, delayed diplotin is called dictyotin stage. The giant chromosomes called as lamb brush. We'll be studying in cell as the basic unit of lipolytin and lamb brush. Here, giant chromosome called lamb brush chromosome is originally decondensed diplotin chromosome. In diplotin, desynapsis takes place. Desynapsis. See the term, diplotin desynapsis. I think it is clear. Now comes last stage of the prophase 1, diakinesis. So dia means across or through. Kinesis means movement. Chiasmata will move towards the end of the chromosome. So that knot will be getting over gradually. So chiasmata move towards the ends of the chromosome. This event is called terminalization. These are mass of following the crucial words. Dictyotin, terminalization, desynapsis, pairing likewise, terminalization. Diakinesis, last stage of prophase 1. So end of the prophase, we know nucleolus, nuclear membrane, etc. disintegrates. That also takes place here. I think it is clear. Now we have to proceed to next page. That is metaphase. So first prophase, most complicated, important. That part is solved. Now remaining comparatively easy. Metaphase 1. So when we enter metaphase 1, it is that means prophase 1 is over. And that will be followed by metaphase 1. Chromosome, now no covering. So chromosome comes to the cytoplasm due to the disintegration of nuclear envelope. So they are free. Bivalence, paired chromosome will be arranged at the equatorial plate, central part. Since each bivalence have 2 chromosomes, 2 centromeres of the each bivalence will arrange as 2 rows at the equator. That is why 2 homologous chromosomes. Mitose only 1. Okay, so only 1 metaphasic plate. Here we can find chromosomes in pairs. So 2 centromeres for each bivalence will arrange as 2 rows at the equator. That is why 2 metaphasic plate, double metaphasic plate in metaphase 1 of meiosis. Is that idea clear? Now comes anaphase 1. Anaphase 1, homologous chromosome separates. The event is called disjunction. If it is absent, it is called non-disjunction. And that is one of the main reasons where Down syndrome is caused. That you will be learning in second year genetics. Now, after separation of homologous chromosomes, separated chromosomes are called univalent with the 2 chromatids. That is why it is called dyad, 2 chromatids. Separated chromosome will move towards the poles. And one more important point, Mendelian factors separate or segregate in anaphase 1. Reduction of the chromosome number also takes place in anaphase 1. I think anaphase is clear. Now we can proceed to telophase. Telophase is the final stage of the meiosis 1, where we can find spindle fibers completely disappear because function over. At the end of telophase, we already studied mitosis, the disintegrated part earlier. At the end of the prophase, that all will come back. So nucleolus, nuclear membrane reappear, chromosome uncoil and elongates. I think idea clear. Now comes cytokinesis. So after karyokinesis, cytokinesis is the next step will take place. 2 daughter cells will be formed. Now here comes a slight variation from mitosis because mitosis only 1 division. Here 2 divisions are there. So that is after cytokinesis, there is a stage may be or may not be. Interkinesis or intrameiotic interphase. Here you can find intrameiotic interphase. This stage is between telophase 1 and prophase 2. So originally it is a gap between meiosis 1 and 2. This is a peculiar stage where no replication of DNA because second meiosis is mainly to reduce the amount of DNA. So no need of doubling of DNA. So no replication of DNA. Elongated chromosome, but not forming chromatin fibers, not fully extended. In animal cells, dual replication will be there. Chemicals like RNA and protein required. for meiosis 2 will be synthesized. Interkinesis or intrameiotic interface is not a must. It is a metabolic procedure. Now we can enter into meiosis 2. Comparing to meiosis 1, meiosis 2 is very easy, just like a replica of mitosis, even though some slight variations will be there. So basically, it is like a mitosis with the slight variations. So meiosis 2 resembles a normal mitosis. That is NCERT page number 127. It is given. And next is an important item, exam point. Meiosis 2 occurs in haploid germ cells. Meiosis 2, haploid germ cells. Meiosis 2 divided into prophase 2, metaphase 2, anaphase 2, telophase 2. Then after that, cytokinesis. Now comes prophase 2. It's easy. It is comparatively simpler than prophase 1. It is short phase. Here no bivalence nature, only univalent. So just like a mitosis. We can proceed to next page, univalent condition. Chromatin become more and more condensed. Nucleolus, nuclear membrane disappear because prophase end. Now next is metaphase 2. At this stage, chromosome in the sense univalent. Previously it was bivalent. Align at the equator, on the equatorial plane, the microtubules from opposite poles of the spindle get attached to the kinetochore of the sister chromatids. Now sister chromatid, previously chromosome. Next comes anaphase 2. Here splitting of the centromere of each chromosome, which was holding the sister chromatid together, by which sister chromatid moves to the opposite pole. That is, area looks like mitosis, opposite pole of the cell. How? By shortening of the microtubule attached to kinetochore. So it will be shortened, so chromatids will be separated. Now last stage, it is called telophase 2. Ends with the meiosis, ends with the telophase 2. That is what given again in NCERT page number 128. Here chromatid reach the poles and they start uncoiling. Degeneration of spindle fibers. Since functions are over, no need. That is why they are all degenerating. Disappeared structures like nuclear membrane, nucleolus will reappear. And remember, 4 nuclei haploids are formed. Then cytokinesis, forming tetrad, 4 cells. Diagram I will put in the last. Cytokinesis can be 2 types. That part not directly mentioned in NCERT, but when I refer some old papers, these 2 terms given. That's why I given it. One is successive, one is simultaneous. Successive means after every nuclear division, cytokinesis will occur. Second one, simultaneous, means here cytokinesis takes place only after the end of both the division. Simultaneous. I think I did clear. Now, significance of meiosis. Many areas we'll discuss till gamete formation, maintenance of chromosome number, variation, mutation, evolution. In connected with the NCERT, there is a famous statement. Specific chromosome number of each species is achieved across the generation in sexually reproducing organisms. Even though the process per se, here you can find a term per se. Per se means by itself. Paradoxically, results in reduction in chromosome number by half. So this line is an important one, paradoxically. NCERT page number 128, reprint edition, March 2024. Then comes NCERT diagram, clear diagrams you can find. Bivalence, all this you can find here. Okay. Then 2 cells will be formed. So chromosome number of at each chromosome with the double amount of DNA. All NCERT figures. Then meiosis 2. So the 2 cells are shown here. Like a mitosis, finally we'll be getting 4 haploid cells. I think it is clear. Now, as usual, here stages can be put in exam point of view, either theory or entrance, identify the stage. How you can distinguish mitotic, metaphase, and meiotic metaphase 1 or metaphase 2 this way. Next, that aspect we'll discuss when we do the questions. Now, as usual, 5 theory question, 5 NEET model question with the key given here. So what all things we learn, we can just pass through it. So chapter 10, that is cell cycle and cell division 3. One more needed where I will give certain areas that cannot be discussed in this episode because it will be too lengthy. Introduction, meiosis, significance, questions. Meiosis, the root term, then scientist. After that, meiosis begins. then 2 division, meiosis 1 and 2, equational reduction, etc. And why the need, or what is the need for second meiotic division, then meiocyte, then meiosis 1, meiosis 2 stages. After that, we entered into each stage. Prophase 1, most lengthy, leptotin, thin-threaded stage. Here you can find bouquet stage. Then zygote, meaning of the term zygote, that is bivalence, homologous chromosome 2 in number, 4 chromatids, 2 centromere in a bivalent. Then sister chromatids, synapsis, synaptonemal complex, etc. Otherwise called syndesis. Then we entered into pachytene, thick. That is the meaning of the term pachytene. And this is the area where we can find tetrad condition will be clear, visibly very clear. Then causing variation through an event called crossing over that takes place between homologous chromosome non-sister chromatids. We explain sister and non-sister, homologous and non-homologous. Crossing over takes place between homologous chromosome non-sister chromatid. Then comes a diplotin. Diplotin means double. Chiasma will be clear. It is X-shaped. Full separation, that is not complete. Certain area it will be linked, that is called chiasmata. Then developing a corrosate, dictyotin stage, delayed diplotin. Then giant Lamprecht chromosomes. Originally decondensed diplotin chromosome, etc. Then diplotin desynapsis takes place. Then diakinesis, chromosome separates, that is terminalization. Then at the end of the prophase 1, nucleolus, nuclear membrane all disintegrate. Now metaphase 1 AC, 2 metaphasic plate, double metaphasic plate because homologous chromosome. Anaphase 1 disjunction. Then non-disjunction leads to Down syndrome, univalent with a 2 chromatid, that is diad. Mendelian factor is separated in anaphase 1, reduction chromosome number in anaphase 1, then followed by telophase 1, final stage of meiosis 1, spindle fibrous couple disappear, nucleolus, nuclear membrane reappear, and chromosome uncoil and elongate. Then cytokinesis, where we can find 2 areas will be formed. After cytokinesis, certain cases, interkinase or intrameiotic interface, not a must. Then meiosis 2, followed by prophase 2, simple, no bivalent nature, only univalent. Chromosome become more clear. That is more clear. Chromatin condense, nucleolus, nuclear membrane disappears. So chromatin, not chromosome, sorry, chromatin become more and more clear. Condensed, nucleolus and nuclear membrane disappear. Metaphase 2, here we can find single metaphasic plate, univalent chromosome, kinetochromatic sister chromatid will be attached. Then anaphase 2, splitting of the centromere of each chromosome. Then anaphase 2, chromatids will be moving. Telophase 2, that is meiosis ends with the telophase 2. Chromatids raise the poles, start uncoiling, etc. And we'll be getting after telophase 2, 4 haploid nuclei. After that, cytokinesis, tetrad of cells will be formed. And cytokinesis can 2 types, successive, simultaneous. Successive means after every nuclear division, cytokinesis occurs. Simultaneous means cytokinesis takes place after end of both the division. Then significance of meiosis. Then after that, diagrams. Then 5 theory question, 5 NEET model question with the key. I think it is clear. If you like this episode, subscribe or like it, share it, comment on it. We can meet later with another episode. Gradually, I will be putting questions also and discussing it. 5, 6 questions and that will be discussed, NEET type question. Time we can wind up. Thank you.